far? Now we got the electron transport chain. It's probably the busiest thing over here. But you understand that it's not one explosive reaction, but we have a bunch of reactions occurring. This is all happening with molecules that are embedded in that inner membrane of the mitochondria. So we have this electron transport chain that is going to use oxygen as its last piece over here to accept those electrons to make water. This occurs in the mitochondria cristae. The cristae is just a fancy word for that undulating area of the mitochondria. We need oxygen here. That's the one that's going to accept the last. That's the last part that's going to accept those electrons, those hydrogens. What's actually going to make ATP is something called ATP synthase. Again, ASE, it's an enzyme, but it's like a pump. So two electron carriers, the so-called NADHs and the FADH2s, those are carrying electrons. They're the ones that are going to convert and change that ADP to ATP. As I told you before, you get 3 ATPs for 1 NADH. And you get 2 ATPs for every FADH2. So the electron transport chain is made up of 4 proteins, or 4 protein components, and 2 things called cytochromes. The cytochromes have an iron in it, which is another reason why you've got to have iron in your diet, right? Big picture. And it's embedded into this, the cristae, the inner membrane of the mitochondria. The carriers will alternate, reduce and oxidize, and accept and donate electrons. That's what's happening over here, step by step. Accepts, donates. The last thing that's going to happen here is that it's going to donate the last 2 electrons to oxygen. And that's going to make water. It's the final electron acceptor. Why you need oxygen. Otherwise it just builds up and has no place to go. You need the oxygen to accept those. Okay, so this is what it looks like. These are basically your 4 molecules, labeled as 1, 2, 3, 4. This is going to be your cristae. You're familiar with that membrane over there, phospholipid bilayer, but that's the cristae. So that's going to be your, so there's another membrane up here. The outer membrane of that, right, if I'm doing this, let's see if we can visualize this. You have your mitochondria. The outer membrane, the inner membrane. The inner membrane is also referred to as the cristae. You have the inner membrane space and the matrix. The citric acid cycle takes place in the matrix. Where all of this is embedded, it's going to be embedded in here, in the cristae. But there's also going to be, which is not drawn up there, I'll show you on the next slide, there's going to be something over here that is going to make ATP. That's your synthase. What's going to happen here, if you can visualize this, This citric acid cycle is making the NADHs and the FADH2. They're making them over here. These items over here are going to pass through these items here and put those extra electrons out into the intermembrane space. The more FADHs that are here, the more NADHs that are happening over here, the more hydrogens get pumped out of there. And it goes step by step, boom, boom, boom. For me to hand something to Melissa back there, if I hand a paper to Samantha here, she hands it to Derek, she hands it to Jesse. It's that thing. Otherwise, I could just work there, but then I'm going to use too much energy to do that. It's a step-by-step thing. This leads to this leads to this, and all this, when you start powering and pushing more hydrogens out here, you're creating, and that's how we're getting back into this, you're creating a gradient. That there's going to be more hydrogens out there than there is in here, and this pump, the hydrogens are going to go through here, and this is where this, and that's the synthase. You're going to take the ADP, and when a hydrogen comes over here, it powers it to make ATP. I have a little video to show you too, so you see this all happen. With the NADH, see, it makes the hydrogens here. Eventually, the hydrogens are going to be pushed out there. If you've got NADHs over here, you're going to get 3 hydrogens out there. If you have FADH2, you get 2 out there. Each hydrogen that comes in is going to make an ATP when it comes back in. So electron transports, we're pushing more hydrogens out there, step by step. They're going to come back through here. There's your synthase. When it comes back in here, the ADP is going to bind with another inorganic phosphate to make ATP. But it has to have a hydrogen to do that. So you got to visualize this whole picture coming through here. The more hydrogens you put out there, you've got this concentration gradient. It's going to go from a high concentration to a low. Right? Things we learned about before. So this thing here is called ATP synthase. Synthase because it synthesizes ATP. Makes ATP. That's why they call it synthase. And just hydrogens just get powered in there because it's simple diffusion from a high concentration to a low. This process where there's a hydrogen gradient going from high to low, we also have a special name for it called chemoosmosis. Chemical osmosis. In this case, the hydrogen osmosis. Not osmosis with just water, chemo. Or heat. All right? Yeah. So yeah, because this thing actually will, this Q part over here actually moves. It's too complicated. It's biochemistry. But it's going into 4 different molecules with a couple of cytochromes, the Q and the C up here. But it's doing all this, it's a step-by-step thing. It would be nice if you're thinking about it, that if there's only going to be one thing to push it out there, but doing this way, NADH happens here, the FADH2 happens over here. So that's why you get 2 out of FADH2s, because it happens further along. you're only going to get one here and one here. Does that make sense? Whereas NAD happens much earlier, so you're going to get 3. You're going to get a little bit more than FADH2. Because they're used, the NADH is used in all of them, whereas the FADH2 is only used in the third one here. And it's pushing all those hydrogens and creating a higher concentration of hydrogens there, which is then going to fuel and push it into this ATP synthase, allowing the ADP to create ATP. All right? Just another picture showing that step-by-step kind of thing. But it's kind of like this, where, like, it kind of shows you, like, passing the paper to Melissa. I mean, I could do this and then walk all the way to the fire, but it goes step by step. There's an oxidation, reduction, and so forth. That's all that's happening over here. Instead of one explosive thing, it's more controlled. And that's what I'm trying to show you here.
Most of the energy harvested from organic molecules during glycolysis and the Krebs cycle is stored in NADH. NADH and FADH2 molecules give up their high-energy electrons in the third phase of cellular respiration, electron transport and ATP synthase, where most of the cell's ATP fuel is produced. The electron transport chain is an array of molecules, mostly proteins, built into the inner membrane of the mitochondrion. NADH gives up its high-energy electrons to the first complex in the electron transport chain. The electrons move from one member of the chain to the next, giving up their energy as they are pulled from NADH toward oxygen. The energy given up by the flow of electrons is used to pump hydrogen ions from the mitochondrial matrix into the intermembrane space. Oxygen captures the electrons in the very last step in electron transport. The last complex adds a pair of electrons to an oxygen atom and 2 hydrogen ions, forming water. The electron transport chain has used the energy of moving electrons to pump hydrogen ions into the intermembrane space. This buildup of hydrogen ions, like water behind a dam, stores the potential energy that was originally in the bonds of glucose molecules. The backed-up hydrogen ions give up their energy when they diffuse through a special protein complex in the membrane called ATP synthase. As hydrogen ions flow down their concentration gradient, ATP synthase captures their energy to make ATP. Under normal conditions, almost all the ATP produced in the process of cellular respiration is manufactured by the energy that electron transport provides to ATP synthase, about 34 ATPs for every glucose consumed.